Well, well, it was great that Sandra gave a very good introduction of both phase separation and aggregation of the protein. And I'm using this very old slide that, well, I would say that is beginning of the 2000 years that we used to all the time think that, well, most of the proteins that aggregate, they is some misfolding pathway that occur for some proteins. In fact, we have learned in the last 10 years, 15 years that many proteins and undergo aggregation and causing disease, at least 50 different diseases. And our lab has worked in with many proteins, including prion protein, the alpha-synuclein, the p53. We now focus today on p53. More recently, we have learned, and also gave a very nice presentation showing that while aggregation that we see in vitro is quite more complicated, and we turn into the cell milieu, and we understand, we have understood a little that it's part of something that's functional that goes wrong, especially because and also Sandra showed very clearly the importance of liquid phase separation to form membraneless organelles, and to especially in the nucleus that I would say probably most of the important events that occur in the nucleus depend on phase separation. And it's just to, and again, we know that phase separation, of course, is functional, but eventually something goes wrong, and there is a gel-like phase transition or a solid-like phase transition. We have seen that with several proteins. We see later more examples of these. But one thing that has been clear is the participation of some polyanions, especially RNA and sometimes DNA. And we know that for some reason, and I want to use this slide that is a review that I wrote with Rama about this good and evil side of most proteins, and then especially for nuclear proteins, and then eventually we have a disease, and if something that is if the cell cannot control the fate of aberrant phase transition or aggregated protein, we have a disease, and that's the story of with cancer. Our lab in the final and the end of 1990 and beginning of the 2000, we're working with different proteins, especially with p53 and the prion protein, and of course they share some characteristics. They have both have intrinsically disordered segments. Of course, the p53 is a much more complex protein. Of course, they both, especially p53, that's a transcription factor, bind the DNA, and the story started. We found that also it aggregates to amyloid or initially amyloid oligomers and fibrils. Of course, when we compare p53 that's related to cancer is a tumor suppressor protein that is very important to control the cell cycle. But if something goes wrong, especially if you have a mutation and there is a lack of a function or loss of a function of the p53, and that can help drive or participate in the pathophysiology of cancer. And in fact, mutations in p53 are present in half of all human cancer, a little bit more than half. And what's curious is that, well, of course, this is a somatic mutation, so you have only one gene that is mutated, but for some reason that was known that the mutated p53 affect in some way the wild type in a way that they're giving this dominance negative effect that people start to think that was a prion-like effect, and that would contribute to to turn the disease to make it faster and to make worse. Anyway, our lab has approached protein aggregation with different techniques, fluorescence, NMR, and especially using also high pressure techniques to go through all these intermediates. And of course, there are shared intermediates with formation of functional oligomers and formation of amyloid fibrils. And high pressure is quite interesting because you can go through this with and it's quite reversible in a sense that you can release the pressure and look at the reversibility, if there is reversibility. And we have observed there are several examples of proteins that we can study, for example, dissociation of amyloid fibrils using high pressure. So we decided to use this technique with p53 that I will be talking, especially because, as I mentioned, we have been very successful in the last 25 years of using high pressure to study the dissociation of fibrils, vitals, and even the denaturation of proteins. And especially because it's, as I mentioned, a unique technique because you can, in a different way than, for example, denaturing, to promote the denaturation, and use techniques, for example, high pressure MR to look what's going on, and you can go through all these intermediates, especially partially disordered states that are populated when the protein goes from folded to fully unfolded and vice versa in the way back. So in 2003, while we published these two papers that curiously using just the p53 is a tetrameric protein, so and you have the DNA binding domain that is most of the mutations, 97% of the mutations that are involved with cancer are in this DNA bind domain that has a single tryptophan. So we decided to study this DNA bind domain by that time, Danielle Shimaro. And curiously, using pressure, usually for monomer proteins, you need much higher pressure to cause the denaturation, and we found very easy to denature. And more curiously was the fact that there was aggregation concurrent with the denaturation, and when we compared, for example, wild type of a mutant that's shown, for example, just by high temperature, showing that the mutant not only aggregates but forms large aggregates with bind of thioflavin T and higher light scattering values. And then Danielle decided to look in the microscope, and the surprise was that some oligomers were found, and especially fibrous, typically amyloid fibers were found. So of course, in this paper we use we use high pressure to shift equilibrium toward this partially folded state that help to go to the aggregate state. And then we proposed at that time that because of that maybe this would explain the negative dominance effect that occurs in tumor cells if you have a co-aggregation of the wild type with the mutant one would have the lack of function and the dominance negative effect. And this was clearly proved in vitro, yet in vitro, was looking the aggregation of the DNA bind domain as a function of time, just at atmospheric pressure at 37 degrees, and clearly the wild type DNA bind domain, that's bind of tyrflavin T, die, and aggregates much slower, and these what's called a hotspot mutant aggregate much faster and with much higher values of tyrflavin T. We looked also the bind of Congo red, and in vitro these aggregates are quite heterogeneous. One can isolate fibrils, and we looked at the X-ray diffraction, and indeed they had this typical fingerprinting of the fibrillar structure. And then the next question was to investigate whether this mutant that is more prone to aggregation would see the aggregation of the wild type. So that was a very simple experiment that we did some years ago, a decade ago. When you have the wild type, it aggregates much slower. But if you had seeds, small, about 2–5% of seeds, we have a much faster aggregation, and showing that in vitro there was this kind of a prion-like effect of a p53. Then, of course, the next question was, oh, okay, that is in vitro, you can show that, but how about in vivo, or maybe correlated with some tumor cells or tumor in cells in p53 in cells from biopsies, for example. So we used an antibody against amyloid that recognize any amyloid structure. In this case, we use A11 that they recognize amyloid oligomers, and clearly shows here that, of course, this is a protein soluble wild type, and the mutant, we don't see any signal, any binding, and when it's in the aggregated state, we have the signal. That's just we use transthyretin bought from Deborah's Fogler's lab, and that showed that it recognized. So we decide then to go to not only to tumoral cell lines, but also to biopsies. And clearly, when we have antibodies against p53, so here by this figure showing the fluorescence of antibodies against p53 and against oligomer, showing in the case of this case of a biopsy that had a lot of that was present, this R248Q mutation. We had much more signal of the A11 antibody, and that's the colocalization, of course, with limitations of this assay, but showing that we're quite colocalized. And in this collaboration with another group, we looked about 90 biopsies, and we can see that, well, of course, when we had the wild type, we had the fairly little bind of A11, showing little oligomers, and but when we had the mutations, different mutations, especially those mutants that cause more severe disease, we had a lot of signal from the A11, from the signal from the biopsies. And this is also clear in another kind of, that was breast cancer, that we have this both in cells and also in biopsy of breast cancer. And the same we find, for example, both in biopsy, but also in tumoral cell lines from glioblastoma cells that we see, for example, in this case that we have this cell line that has a mutation, the ME231 mutation. We have, when you compare, for example, with wild type, we have much more signaling of the amyloid oligomer. And in this paper that was developed with different people in the group, including Guilherme de Oliveira and Murilo, that was a graduate student, now as a postdoc. We're particularly interested in this mutation because it causes a gain of function. It turns the cells resistant to chemical treatment. So there is a resist, for example, to temozolide, TMZ. So we're trying to correlate not only the loss of function or the negative dominance effect, but also an oncogenic gain of function, and that was our interest. Again, when you compare the speed of aggregation, this mutation, that's the isolated DNA bind domain, it's much faster, aggregate faster, and with in much higher quantity. That's shown by diflavin T and also by light scattering when compared, for example, with the wild type. And in this particular work, we're interested, okay, we can see when we fix the cells and we do the histo microscopy and treatment with antibodies, but we're interested in watching if in the cells this would occur, and for that Murilo used some new cells that were transfected with the EGFP p53 and compared wild type and mutant, and clearly and using fluctuation microscopy. That's also collaboration with Enrico Graton that I spoke yesterday. We showed that in this case, when compared with the wild type, we had presence of oligomers in the nucleus of the cells. So the story start to get, especially in the last 6-7 years, that indeed the aggregation of p53 of mutant p53 is a key event in the pathophysiology of cancer, and not only this kind of a prion-like effect inside the cell, but also eventually also in transmitting this phenotype to adjacent cells and have the cancer progression. So that's a lot of study in progression yet. And here just to show a few studies of different groups, particularly showing that not only p53 aggregates with white mutant to white type, but also with the p63, p73, and then people start to look ways to prevent that, and also more recently the interaction not only of with proteins involved in tumor suppression, but also, for example, in Alzheimer's disease co-aggregation with tau. So it turned out that indeed the aggregation of p53 could be a therapeutic target. So we decided to look for possibilities of blocking this aggregation, and it's quite important, of course, that we look for ways to prevent the aggregation, and it's not only aggregation of p53 or more typical aggregation, but also heteroaggregation of other factors. As I mentioned, p63, p73 are tumor suppressors, and curiously, it's quite similar to p53, but it aggregates much slower, and we probably because it's a little bit more stable. And in this paper, we try to correlate with a lower exposure of backbone hydrogen bonds. And also there is this correlation that, again, looking by high pressure, that p53 and p73 are much more stable against pressure. So we decided to look for possibilities of preventing the aggregation using some compounds. I want to go through all the compounds. We have tests just to give some examples. We start with resveratrol and resveratrol derivatives. We know that the resveratrol is present in grapes, in grape juice, in wine, and that was a work done during the PhD of Daniele, and she found that indeed, with other students, that resveratrol can inhibit aggregation. It inhibits aggregation of mutant p53 at the lower concentration. Fortin is still at very high concentration that would be equivalent of having 10 bottles of wine per day. That's probably not health. That is shown not only in vitro, but also looking in the cells. And so again, here in this part of the slide, one varies the concentration of resveratrol, and one can see that the aggregates that are found, for example, without resveratrol, they tend to decrease at least about 50%. And also in xenographed animal models with the breast cancer cell line, also there was a great decrease, and showing the possibility maybe of using this molecule as a starting point to have a modified resveratrol, and showed here, for example, that inhibits cell proliferation, inhibits cell migration, and that's what we propose. So in another molecule that we also tried was a molecule that was already known to be an anti-tumoral drug. That's Prima-1, that is known to make in a duct with cysteines of the protein, and we found that indeed the Prima-1 inhibits aggregation, and that is in vitro. That's also shown that the oligomers decrease quite substantially. And in this experiment that was done by Julia Ferretti and Luciana Rangel, was very clear that when we look at we do immunoprecipitation with A11. If we treat with Prima-1, one can see that there is no p53 in the aggregate. So we recover the soluble state of p53. So in that clearly Prima-1 inhibits. We are looking for other molecules, and Julia has advanced quite well. Find, for example, this RCP17 can inhibit aggregation at concentration about 10-fold lower when compared with Prima-1. This is clearly shown here in this experience that we use this antibody that shows the recovery of the folded state of p53 when treated with RCP17. We have complete recovery, and also we have less aggregation in this immunoprecipitation assay, and that's shown both here using different immunoprecipitation or using this assay that reviews the aggregation. Other, of course, investigators are looking for molecules. For example, there is this recent paper that showed ADH6 inhibiting aggregation, and this very nice discovery of not only a compound, but in this case of a chaperone that can convert the aggregate misfolded state of p53 into a native state, even when you have the mutant p53. Maybe James Short will talk a little bit about that in his talk. And the last point I want to stress is that we still are far away from understanding what is going on, because remember, to have this gain of function at some point, these oligomers, don't know if they are in phase separated or not, or we didn't know, but they have to have, they needed to have to bind eventually to nucleic acid. And this more recently we found that similar work that Sarah just present, that p53 can go through these different states. That was work done by different people in the lab and with Guilherme and different postdocs, and we could find that it transits from liquid states eventually to gel-like and solid-like states. And we use different mutations. So these are the same mutant of glioblastomas, the brain tumor. This one is a mutation from hepatocarcinoma. And then they both aggregate much faster. So when we look at the in vitro, by the use of some crowding agent, we could see that we have these beautiful liquid droplets for both wild type and the mutant, in this case the M2T7I. And but clearly already both, we don't have full recovery of any of them in vitro, but of the DNA bind domain, but clearly the mutant one is completely in a solid state, and there is no recovery. But the main point of this work was to use again back the cells transfected with the mutant p53, the full length protein. And in this paper, we could see that most of the fluorescence, both for wild type and for the mutant, is concentrated in the nucleus, and they seem to be in this liquid state, because when we use a laser pulse, we can have a full recovery for both of them, a little bit less with M2T7I. But when they are particularly in some nuclear bars, especially in the nucleolus, the M2T7I recover much less. So in that, again, probably in there that eventually having this gain of function effect, you have maybe because of the presence of polyanions there, they gain of function, they convert to this aggregated state. So here just show that in vitro RNA turned the aggregates very fast to the droplets quite fast to the aggregated states. Heparin, that's work with 20, influence of 20, showed that in the other way around, heparin tends to, in vitro, that was done in vitro, of course, convert it to this droplet into a state that is less solid and probably more like a gel-like, and recovers after the photobleaching. This paper also showed that some condition that partially unfolds the p53, like guanidine, leads into liquid states, and at this condition we could revert with a high pressure. I won't go through this data, but clearly, just to summarizing, that we were able to go a little bit through some of these states. Still, as I mentioned, we want to have probably more molecular insights how p53 in this oligomeric state are giving, going to having this gain of function characteristic. So there is a lot of things to be learned. So I think most of the conclusions I have go through. We do believe that the aggregation of p53 or the phase transition of p53 is a marvelous target to for uh, for cancer therapy, and particularly we do need to understand more about this transition between liquid state to gel solid state to make probably better drugs. It goes in the same direction of the prion protein, and I just want to finish saying that we think, well, that's quite new, phase separation, but gut, the famous writer and poet that was he was a chemist. He's in book Electro Finite. He was already thinking about phase separation, and maybe Shorten was also thinking about that without saying, but because it does need to use physical tools to understand that, and want to once more to acknowledge all the people that did the work, finish here. Well, while a lot of old collaboration had this, that Enrico was present in this celebration of 30 years of my lab, but many other commemoration. And once more, thank you very much. I think I interrupt here.